It's a real pleasure to be with you and to be thinking about how we can use plant science to address some of the global challenges that we face. Very nice of you to bring some floral tributes, actually, although it's not actually necessary. Yeah, bring booze next time. So I want to start by thinking what plant science has to offer for these global challenges, and I'm going to focus on food security and climate change, two of the biggest issues we currently face. So the challenges are quite severe. This is an astonishing and slightly terrifying statistic from The Economist. We need to produce more food in the next 40 years than in the previous 10,000 combined. Now that assumes that food waste continues at the same level and meat consumption continues to increase and so on. But we certainly have a food production challenge, particularly, of course, in the face of climate change. And climate change is going to interact with this challenge of producing enough food and could make it very substantially worse. So 1 metre sea level rise could flood 17% of Bangladesh, and that's one of the world's poorest countries. And of course that will have a severe impact on the production of rice, reducing the rice farming land by about 50%. And rice, as I'll talk about later on, is not very tolerant to salinity and saltwater intrusion. And it's not just that climate change will impact on agricultural production, because agriculture is indeed part of the problem when it comes to climate change. So agriculture is very resource intensive. Uses about 70% of the world's freshwater resources, it uses a third of our land resources, and it's responsible, depending exactly how you calculate it, of at least a quarter of our greenhouse gas emissions, as well as contributing to deforestation, which again causes more climate problems. So I hope you can see that all the challenges that we face are somewhat interconnected, and it's sometimes known as the nexus, the food, energy, water and environment nexus, a sort of sometimes called a wicked problem. So something that's very entangled and difficult to address. And this is Ban Ki-moon, former UN head, who's talking about how we need to connect the dots between climate, poverty, energy, food and water. We can't address those issues in isolation. So one of the themes of this talk is about how we tackle these complex problems using plant science, but interacting with other disciplines. And that's the day job for me. At York, I run an interdisciplinary research institute called the York Environmental Sustainability Institute, far too much of a mouthful, known as YESI. And we build interdisciplinary partnerships to try and look for sustainable solutions to these kind of global challenges around food production and climate change. We've got 3 research themes in the Institute. So one of them is around sustainable food, and I'm going to talk on around the production side, but we also look at the resilience of supply chains and consumer behaviour. And we look at resilient ecosystems, particularly the impacts of climate change, but also ecosystem services and conservation. And we have a theme around urban living, pollution, and the impacts of large-scale urbanisation. But I'm not going to talk about that today. I want to focus instead on crop production, food production. And I want to think about, first of all, why our current crop production system is in trouble. And one of the reasons is the losses to pests and diseases. So here are 3 of our main crops: rice, wheat and potato, at least round this region. East Yorkshire and Vale of York, potatoes and wheat are big crops. Don't grow a lot of rice in Yorkshire, it has to be said. So if we don't use any pest control on these crops, on average, we lose between half and 3/4 of them to pests and diseases, either pre-harvest or post-harvest. And even with our best efforts of mechanical, biological and chemical control measures, you can see we're still losing a lot of food production to these issues. Now, we've got two problems, apart from the sheer scale of the losses. One is that a lot of these control measures are currently unsustainable. They involve toxic chemicals, which are likely to be banned or have lost their effectiveness. And the other problem is that actually, if we could just change some of these losses by not very much, we could feed an awful lot more people. So it's a key issue, and one where we're struggling a little bit because our crops have been selected for the things we want from crops: growth and yield, unsurprisingly. So, does anybody recognise these delightful looking objects? One of our favourite food types. Potatoes. That's what wild potatoes look like, and this is what Sainsbury's potatoes looks like. So, of course, what we've done is made them all lovely, smooth and uniform, much bigger, and they've lost this red coloration, which is chemicals that protect them against pests and diseases. And so wild potatoes, for example, do not suffer to anything like the same degree for potato blight, for example. So we've removed those chemicals and we currently fill the void, as it were, with artificial ones. So we can't keep doing that. So what can we do instead? Well, my background is in plant ecology, so my instinct is to look at what happens in the natural world, such as it is, but in plants, wild plants. What do they do? How do they stop being eaten by pests and diseases? So for many years I worked in Scotland on heather moorlands, and they're heavily overgrazed. So here's our little patch of heather which is flowering, and all the overgrazed heather outside isn't. And the canopy tends to be destroyed by the heavy grazing, and you get these grasses, this one in particular, Narda stricta. And I thought, well, why is it that the sheep and the deer and everything else does not like to eat Narda stricta? And I thought, well, maybe it's got some toxic chemicals in, and I spent ages looking. And then I spoke to a colleague who was at the University of Manchester, who's now at the University of Hull, Roland Enos, and he said, well, it's because of all the silica. And I said, the what? Hadn't thought about that at all. But Nardus contains a lot of silica, and it's deposited in these spines and in these granules and in these little razor blades like this. So unsurprisingly, herbivores don't like it very much. So I thought it would be interesting to investigate this kind of defence a bit more, because it's been rather overlooked. So I'll tell you a little bit more about it. So silicon is very abundant in soil, second most abundant element. It's not always very accessible to plants, but when it is, some of them can take up a lot of silicon. So it dissolves in the soil solution as silicic acid. It taken up into the plants and deposited, and then it, through senescence and herbivory and decomposition, returns to the soil. And that's a slow process. And as I say, it's deposited in these little granules and these spines. It fluoresces, so you can see it quite easily how it's deposited. And it's very abrasive. So it's a bit like eating, getting sand in your sandwiches. So if you've ever been down the beach in this lovely hot weather and you've dropped anything in the sand and brushed the sand off and tried to eat it, it's that kind of feeling. It's very abrasive. So what do herbivores make of silicon? Well, this was our first sort of experiments in this area a long time ago now. And we looked at 5 different grass species: Poa annua, Lowland perenni, Festuca ovina, and Agrostis capillaris and Brachypodium pinnatum. And these were natural native grasses that differed in their sort of palatability, and we wondered if that was due to silica. And we reared them in low silica soil and higher silica availability. And you can see that two crop pests we tested them on. So this is armyworm, Spodoptera, and locusts. and this is the amount they eat from silica-rich and silica and plants low in silica. And you can, sorry, the other way around. I'll get this right soon. So that's low silica plants. They eat a lot more than from the high silica. Both species do that. So basically, silicon was quite an effective feeding deterrent. But when we tried it with another crop pest, this is a grain aphid, Titobian, it wasn't anything like as effective. The aphids didn't seem so bothered, and in fact, in one case, they even seemed to prefer the high silica plants. So our conclusion early on was that silica was effective against herbivores, but more so against foliar feeders than against aphids. And at the time, we didn't realise why that was. We do now, so I'll tell you about that later on. But of course, it's not just defending against pests through deterrence. We want to see if it actually has an adverse effect on the herbivores. So this is relative growth rate, and our Spodoptera, again, our armyworms, and 3 different grass species. Well, these two are the same, but another one. And again, we've got our high silica plants, dark bars this time, no wonder I keep getting confused, and our low silica plants. And you can see that the growth rate differs between the different species, but it's always consistently lower when the insects are forced to eat high silica plants. And the reason is that they fail to absorb as much nitrogen from the high silica plants. And nitrogen is a limiting nutrient for most herbivores because there's far more nitrogen in animal tissue than there is in plant tissue. So why were they failing to extract nutrients? Well, a great thing about working with insect pests is nobody cares if you chop their heads off and look at their jaws under the microscope. And insects that feed on grasses often have quite large heads, not for their brains, but for the jaws that they need to cut through plant material, which is quite tough. And so when you rear these insects on the low silica plants, this is what the jaws look like. You can kind of see these ridges, these cutting grooves. When they've been feeding on a high silica plant, you can see that their jaws have been completely worn down. And remember that insects molt, so this process is very rapid. So this happens in as little as 48 hours. So, and this is our measure of mandible wear. And you can see, again, it's different on the different grass species. So they wear quicker on Deschamps than they do on Lolium, which is a more palatable species. And they always wear quicker on high silica plants. So we felt we had quite a good grasp of the system. Silica is an effective defence, deters feeding by making the plant very abrasive so the herbivores can't wear their teeth down and can't cut into it properly and can't digest it and can't extract the nitrogen and so they grow slower. So that all seemed very straightforward. And when we plotted silica concentration in different grass species, this is Poa, one of the ones I was talking about, this is Nardus and Deschampsia right at the top here, there's quite a nice correlation with abrasiveness measured as the depth that grass leaves can cut into a perspex plate. So Roland, for reasons that I never fully ascertained, had a machine in his lab that measured the wear on women's clothing, and he used that to sort of test how much the grass is cut into a perspex plate. And we could correlate that quite well with silicon. Except that when you looked at, say, these two species, there's Festuca rubra and Festuca ovina, and they're quite closely related species in the same genus, and they had similar silica concentrations. But Festuca ovina is much more abrasive than Festuca rubra. So it's more than just how much silicon a plant has, it's what it does with it. So more of that in a moment. But we found that that was a very good explanation of the effect of mandible wear. So here is Festuca ovina with a very low silica concentration, causing just as much mandible wear as our Lolium here with a much higher silicon concentration. So there's something about what plants do with the silicon they acquire that determines how good a defence this is. Now, the other thing we discovered, which was a big surprise because physical defences like silicon were not thought to be inducible, but they are. So this is an experiment we did where we damaged plants either not at all, controlled bars, with scissors, these bars, or with locusts again here, and then we thought we'd have some cuter herbivores, voles. We didn't chop their heads off, I hasten to add, and that's the dark bar. Now, two things strike you about the change in silicon concentration after damage. Firstly, that a single damage event does not induce a change in silicon uptake. Plants have to be damaged repeatedly and above a certain threshold before they take up more silicon. And the other thing that you'll notice is that induction takes place with mechanical damage, things like scissors, but it's much greater with real herbivores. And that is not something that's a surprise. Plants are very good at detecting when they're being attacked by real herbivores, and they do that through things like cues in the saliva of herbivores, for example. I did a great experiment as a PhD student when I took caterpillars and squeezed them like tubes of toothpaste so they kind of spit all over the plant surface, and you can show that that's the cue that increases defence allocation. So plants are quite clever, they know what's happening around them, but they vary a lot. As you go through your degree looking at plants, you think, why are they so variable? But actually, variation is interesting because it always tells you something. So this is a study we did with colleagues in Norway, and this is two plant species, Deschamps and Avenella, and we've got here control and clipped plants from different genotypes in these two species. So the first thing that you can see is there's difference between the two species. Generally, Avenella isn't that impressed with damage, doesn't do very much. Deschamps usually makes a big change in silicon allocation. But you'll also see that there are some genotypes within Deschamps that don't bother very much, and there are some genotypes within Avenella that do. So there's within and between species variation. So why is this? This is what we need to understand if we're going to use these kind of things for crop protection. So what we need to know is much more about how silicon's taken up in plants. Now, the slight difficulty is that we still know surprisingly little about that process. And there's a huge debate going on at the moment in the literature about to what extent the plant is in control of this process and has active mechanisms to control it, and to what extent it's a passive process. And the answer probably is somewhere in between, and depending on the particular plant or the environment. But this is what we know so far. And of course, different roots have different structures. So rice has a rather different structure to the roots of barley or maize. But generally what happens is plants have a transporter called LSI1, which is an aquaporin type transporter. So it lets water into the plant, into the roots, and it lets a lot of other things, including silicon. But this one is about the right size for silicon to come in. So that's a passive process, and then silicon flows along through the root and then needs to get into the xylem. And that process seems to be controlled by LSI2, which is an anion pump. It's an active proton pump, so this is an energy-intensive process. And then it sort of zooms up the plant, and there are various controls, supposedly, like LSI6, that direct where it's supposed to go. And there's also LSI3 is also involved in this process. Not quite sure what happened to LSI4 or 5. They never seem to appear anywhere. And then LSI6 sort of uploads it out of the xylem into the plant cells, and then somehow or other, and we have absolutely no idea how or why, it gets deposited in these different structures. Now, the idea at the moment is that this process reflects plant cell wall characteristics and things like callos as a structure, but the evidence for that is developing, but it's still not cast iron. Okay, so LSI1, we know it's important. It was first identified by a Japanese group, by Ma, and he created a mutant. So this is the wild type, this is the mutant with no LSI1, functioning LSI1, rice, and yield is massively reduced, and pest incidence is much greater. So we know that this process is important, and it's important in the field. Now, if we're going to understand how silicon content varies, if we're going to use this process, we need to be able to measure silicon easily. Now, unfortunately, it's actually very, very hard to measure silicon because it's incredibly hard and, as you can tell, why it's so abrasive. So the usual methods were to digest it in hydrofluoric acid and do lots of complicated chemistry. That was a bit of a pain and meant you couldn't do it very quickly. So my lab developed this method using X-rays. So you use X-ray fluorescence, and then you can not only measure how much silicon there is in a plant, but you can actually see, if you connect your X-ray fluorescence machine to a scanning EM, you can see where the silicon is. So this is a barley leaf, so you'll be glad we don't eat the leaves. There is the silicon associated with these big spines. If you cut through the leaf, you can see cells full of silicon. You can see silicon in the tips of the spines and underneath. And this is an undamaged leaf with a bit of a gap here between spines. This is a damaged leaf, and you can actually see increased silicon deposition as the plant is damaged. So let's go back to our issue around Festuca rubra, Festuca ravina and Deschampsia. Remember, Deschampsia was the most abrasive and unpalatable, and these two seem to have the same amount of silicon, but this was much more abrasive than this one. And once you look at the leaf surfaces and the deposition of silicon patterns, you see why that is. So this is Deschampsia. Nobody would want to eat this really, with these enormous silica-rich spines all over the leaf surface, especially at the edge. This is Festuca rubra. So it's depositing its silica all over the surface there. And you can see why it wouldn't be very abrasive. It's only got a few little knobbles, and technical term. And Festuca ravina looks like it's got measles. It directs its silicon very precisely to a particular place on the surface. So what happens when you give these plants more silicon, or when you damage them? So you can see that Deschampsia, actually these are very similar, so I'll put them both up. Deschampsia not only has even more spines, covers the surface with silica, and it produces these odd little structures as well. Festuca ravina does the same, much more silicon deposition, and starts to grow spines, which it doesn't unless there's loads of silicon or it's being damaged. And Festuca rubra doesn't do very much at all, really, just carries on the same. So you can see now why this would be much more powerful strategy against insect herbivores, and so would this, and this wouldn't be that great. So why does Festuca rubra do this? Can anyone think of an advantage? I'm tired of doing all the work here. Can anybody think of an advantage of this strategy as opposed to this strategy? Go on, be brave, someone, shout out. What might that defend against really well? If you're a plant, you're sitting there. Microbes or pathogens, only fungal pathogens, you've got to try and tunnel your way in through that. You're not going to like it, are you? But if you're tunneling your way in through there, you can find a gap. So it may be that plants have different strategies with their silicon for different defense mechanisms. Okay, so how do they control all this? Well, on to more recent work now. So this is one of my PhD students, Emma McLarnon. She was a great PhD student. She was absolutely fantastic. She didn't want to be an academic, so it's far too much hard work, but she was really clever, and she's now gone to work for HMRC to hunt down tax evaders, which sounds like much more fun. So she looked at different varieties of tall fescue. Now, her project was part funded by DLF Trifolium, which you probably haven't heard of, but they're a very famous and very large grass breeding company, and they breed turf grasses and forage grasses and so on. And they had a breeding strategy that meant they had lots of different varieties of tall fescue. And some of them had very soft leaves, and some of them had very harsh leaves. And their method of testing them was to walk through their glass houses and go, ooh, very soft, ooh, semi-soft. Ooh, semi-harsh. Harsh. Very, very soft. And we thought that wasn't terribly quantitative, really. So we thought that it might be something to do with silicon, and we might be able to measure it a bit better for them. So here is a harsh variety, and here is a soft variety, and you can see straight away on these EMs that there's far more spines and surface structures, and they're far bigger on the harsh variety than on the soft variety. And when we look at the silicon uptake as a percentage of root biomass, so this is how efficient these varieties are at taking up silicon from the soil. You see, you thought I was joking. Very, very soft, soft, semi-soft, semi-harsh, and harsh. And you can see that that correlates really very nicely with how well they take up silicon. So actually, it wasn't a bad method, actually, walking up and down with your fingers, but this is a bit better. And then, of course, she wanted to know, well, why does the harsh variety take up more silicon? So she's just looking at 3 varieties now, very, very soft and harsh. And this is leaf silicon, and she's got undamaged plants, which are the white bars, and then these green bars are from damaged plants. Undamaged leaves, the paler ones, and damaged leaves on damaged plants. So the first thing that you can see is that the very, very soft takes up less silicon than the harsh variety, and the very soft's in the middle, which is kind of what we've found before. You'll also see that, that actually damage doesn't have a great deal of effect. The very, very soft doesn't show much induction. Harsh shows much more induction, very soft somewhere in the middle. But you'll see that actually the induction for the very soft variety is not very different between the damaged and undamaged leaves, but actually what happens in the harsh varieties is that even the undamaged leaves show more silicon. So in other words, the induction in the harsh variety is systemic. So it travels even into into leaves that haven't been damaged. So then she looked at characteristics of these varieties, and the harsh variety has more stomata, greater stomatal density, it has greater stomatal conductance, it has greater transpiration rates. So we thought, yes, this is all about transpiration. Effectively, what happens is the harsh variety just slurps up more silicon. It's all a passive process, dead easy. So then what she did, because we're nothing if not sophisticated at York, was stick all the plants in a bag, because that way you make the atmosphere very humid and you make the transpiration rates low and very similar. And then we found something that we completely didn't expect. All the varieties showed very similar levels of silicon uptake, and so we thought, well, okay then, maybe the genotypic differences, the varietal differences, are due to transpiration, and when you make transpiration the same, you don't see any differences in the genotypes. But the real thing that we didn't expect was that we saw induction in all 3 varieties, even the soft one, the very, very soft one, in fact. So what this tells us is that some aspects of silicon allocation within a plant are not controlled by transpiration. The plant has some direction over this. Now, this is still quite controversial. Not everybody thinks this is true. So it's still a matter for debate. But it seems to us, at least, that there are some interesting mechanisms that the plant has. So basic silicon uptake is transpiration driven, but there are other aspects that are not. And there are other studies that have metabolically inhibited transpiration or metabolically inhibited proton pumps and seen that silicon changes. So there's a lot going on, and a lot of it we don't understand. What we do understand is that there's huge variation, as I've just shown you, in silicon uptake. And this is another experiment that we started. I've got a new PhD student now working on wheat. And these are 15 wheat cultivars grown in exactly the same concentration of silicon in hydroponics. So there's no soil, there's nothing different. They're in a little tub, all with the same amount of silicon, left in exactly the same conditions. And there's a huge variation in between the cultivars in what they take up. So this is Mar's work again from in rice. So he's got 2 types of rice here, Nippambare, Kalasath. And he sees that Nippambare has a much faster silicon uptake. And he related that to the relative expression of the LSI1 transporter. So we thought, we need to get to our fescue and look at what happens in terms of their expression of these transporters. Now, the difficulty we had, of course, is the tall fescue genome is nowhere near as well characterised or as easy to work with as rice. But anyway, so Emma took harsh varieties and very, very soft, not damaged and damaged, and took RNA samples from the roots and did all this amazing stuff, which you guys probably know more about than I do. You know, the Trinity pipeline and Edge R. Very technical. Anyway, to cut to the chase, here were the results. So in her undamaged plants, she's looking at the very, very soft and the harsh. She didn't see any differences in LSI1 expression between the varieties. She did see a slightly higher expression of LSI2, that's the root efflux transporter, remember, in the harsh variety. So that might explain some of the differences. And in the damaged plant, she did see some upregulation of LSI2 in the harsh variety, but not in the very, very soft variety. So that does kind of match what she was seeing. Chinese colleagues have also found no differences in LSI1 expression between different varieties, but also seen some differences in LSI2. So it may be that LSI2 is more critical. It's not just the rate of uptake, it's how you then pump it into the rest of the plant that could be really important. Okay, so we know that phenotypic effects like transpiration and damage and silicon content affect tall leaf fescue silicon, so does genotype, so does the transporters. We now know that other defense genes do, so silicon and the jasmonate anti-herbivore defense pathway interact. So there's loads of things that affect how silicon is deposited. But why is this any use? I can hear you say. Well, understanding more about the deposition has helped us with our herbivore issue. So we've now can perhaps understand why foliar feeders are more affected than aphids. You can imagine avoiding aphids manage to avoid, because they're phloem feeders and penetrate the surface, they can probably avoid some of the worst effects of silicon. So that may be why they're less affected. But when we did a meta-analysis on herbivore performance across all guilds, you can see that the fluid feeders are less affected than the chewers and the borers, but overall, herbivores are adversely affected by silica, and pretty well all groups of herbivores show at least some decline in performance when silica is present. So that gives us a lot of hope that silicon could be really helpful in crop protection, because, of course, most of our crops are grasses. Now, we've changed them a lot. We've done an awful lot to wheat over the years. And this was maize in its original form compared to now. So we've done a lot of change to our crops, but they're grasses. So they've got a lot of silicon in them. And there's a lot of interest now in using silicon in crop protection. There's whole books on it. I love this paper. Plant silicon, yeah, disease resistance in plants. Silicon leads the way, and the rest of us are following. There's even a paper out, Silica, the silver bullet for mitigating biotic and abiotic stress and improving grain quality. There is nothing that silicon can't do, believe me. So if we're going to really use this in crop protection, we need to know what are the genotypic physiological environmental processes governing silicon uptake and deposition. So I've already presented some work on that, and I'll show you a bit more in a moment. But we really want to know how to exploit that variation for practical benefits. And the real silver bullet is whether silicon can help protect against abiotic and biotic stresses together. In other words, against herbivory and against drought. Okay, but you're probably already thinking, hang on, there's a problem here. We've domesticated our crops, haven't we got rid of the silicon? Well, we know, of course, that as we go from wild species to early domesticates, we lose a lot of genetic variability, and as we go to modern varieties, we lose even more. And Mayer's study a bit back showed that the biggest change during domestication was the loss of secondary metabolites. So, as we've domesticated our grasses, have we got rid of silicon? No, we haven't. So this was a student of Colin Osborne, colleague in Sheffield, and she worked with myself and Colin to look at the change in silicon concentration from wild progenitors, landraces and modern cultivars across a number of different crops. So we've got barley, rice and sorghum and wheat, so lots of and maize. So there's a little bit of variation. So for some of them, there didn't seem to be much change between the wild progenitor and landrace, but there was a change to modern cultivars. Some of them kind of, it was kind of the other way round. But overall, there was no real detectable effect of domestication on silicon allocation. So we found that it's a very conserved trait. And interestingly, we've the work I'll show you in a moment suggests that that's true, because we find very little variation in LSI1 expression across huge numbers of diversity panels. So it looks as if this is a very conserved thing, which is good. Just want a quick reminder around climate change. This is the FAO's, UN Food and Agriculture Programme's assessment of the impact of climate change on crop yields. This isn't great if you're colourblind, sorry about that. This is, but basically red is bad, green is good. So large areas of the world, particularly ones where food security is already an issue, Africa and India, it looks like crop yields will be very badly affected, whereas the people causing the problem, we're looking at US and Trump, there's not going to be such a big impact. The other issue, of course, is water scarcity. So this is the same UN FAO projections for increased water scarcity, and some very worrying statistics or predictions for India, Africa and Australia. So water stress is a huge issue. Now, silicon I mentioned is there's interest in it being able to help with resilience to drought. Now, there's a number of mechanisms around this. One is that it may strengthen the roots, allowing the roots to penetrate harder soil because they're tougher. It may reduce water loss from the foliar tissue. But whatever the reason, it's certainly true. So those 15-week cultivars I told you about, this is their silicon concentration, and this is their inhibition, their decreasing growth that happens when they're droughted. So some of these varieties with very low silicon show a 90% decrease in growth when they're droughted, whereas ones with higher silicon show a much smaller effect. Now, one other mechanism that my colleagues at York are very interested in, and this is the work of Franz Matthias, is that it seems that for salt and drought tolerance, you need to exclude sodium and maintain high shoot potassium. And again, you can see that silicon and potassium content in the shoots of these droughted cultivars are correlated. So there's something about having a high level of silicon that means that you can take up more potassium. So we've now got a project called Rice Research Initiative, increasing rice tolerance to salinity and drought. That's looking at the role of silicon, that's my bit, identifying candidate genes, testing those through gene editing, and trying to transfer the candidate genes into elite cultivars. So just a little bit about the work we've done so far. It's a new project, we're only 1 year in. And there's 300 different rice accessions from Susan McCucci's collection at Cornell, one of the world's leading rice breeders and rice geneticists. And we're asking whether tolerance to drought and salinity correlate with silicon uptake across these 300 genotypes. And if it does, can we identify candidate genes? Well, the first thing that we've shown is that using our hydroponic system, generally growth rates are very slightly higher under high silicon, and that both root and shoots take up more silicon when you give them more silicon. So far, so good. So nothing new there, but what we then want to do is see if we can correlate those uptake traits with any candidate genes. So this is a genome-wide association study, a so-called GWAS study, which you may have come across already. So what GWAS does is looks at a whole range of genetic variants, so-called single nucleotide polymorphisms, and sees to what extent they are quantitatively associated with a particular phenotypic trait measured across a large number of genotypes. So the 300 genotypes, this is our rice chromosome, and all our different SNPs are plotted along here, their position on the chromosomes, and this is the silicon level. and then you can identify the regions on particular chromosomes and hopefully particular SNPs eventually that are associated with high silicon uptake. And so far we've found about 1,000 potential genes, so that's way too many, of course, so we're homing in on the ones that are particularly important. And they look to be transporters linked to abiotic stress and transcription factors and kinases, but also, as I'll show you in a minute, some genes associated with cell wall function. So that work's ongoing, but we've now got a similar project, and this is dealing with a key problem that's associated with climate change. And that is, how do we produce enough energy as well as enough food? And at the moment, when you grow a ton of rice, you get 1.5 tons of rice straw. So what are you going to do with that? Now, ideally, you'll turn it into energy, but there are some big problems with doing that. One is that rice straw is very high in silicon, so it's not very palatable to animals, so you can't feed it to anybody, and when you try and burn it, it clogs up the furnaces. So what people do is burn it in field. And that's a waste of resources, and it also causes lots and lots of pollution. So here we are. These are rice fields in the Punjab. All these little red dots are NASA's satellite images of rice burning, and this is the pollution from that over Delhi. So it's a huge health issue. This does no good at all for climate change. It's not great at all. And the other problem, this project is based in Vietnam actually. Trying to cross the road in Vietnam is an entertaining experience. And if we can have better fuels to fill these, all these myriad motorbikes, it would be a good thing. And we can also feed our rice straw to caribou. That's water buffalo, not to be confused with caribou. So we want to try and solve these problems by making rice straw able to be used for animal feed and to be turned into biofuels. And at the moment, silicon is preventing both. And we're all admiring here a Japanese-funded project that has got, this runs on rice straw biofuel, not very efficiently, but we're all admiring it. Those are my feet there, by the way. So how can we make this work better? It's a big interdisciplinary program led by Simon McQueen Mason at York. We want to increase understanding of the role of silicon in digestibility, identify rice accessions that differ in that trait, and then we can identify the genes responsible. Then we can understand how we can alter silicon to make straw more digestible for animal feed and better able to produce biofuels. We don't want silicon so low, of course, that the rice falls over, and we don't want silicon low in the leaves in case it gets damaged by insects or pathogens, but we want to be able to understand how to control it in the straw. So we've got another GWAS study, essentially. We've got a Vietnam panel with 172 accessions, and they've been sequenced to a very high level. We've got a Philippines panel with 215 accessions. They're being genotyped at the moment. We measure the silicon with our PXRF machine. We do our GWAS, and then, hey presto, we get some great markers for breeding low silicon. That's the general idea anyway. It's proved a little bit more complicated than that, as you can imagine. But the good news is, this is the Vietnam panel, this is the Philippines panel, and you can see there's a big range in silicon content across the panel. So we have got some diversity to work with, some genetic diversity to work with. When we look at the digestibility in terms of the sugars released, which is what you're trying to do, of course, to make biofuels and also to make it more palatable for animals, we get a big variety, a big range of digestibility in terms of sugar released across the accessions, and this one in particular was very good, very digestible. But when we plotted the sugars released against silica, we get a good correlation. When we plot them against lignin, we do not. So the interesting thing here was that the digestibility of rice straw in terms of its potential palatability to animals and its potential to produce biofuels is much more related to silicon than it is to lignin. So that was our first surprise, but of course it was good news for me on the silicon part of the project. So now we're doing GWAS. So we've done a preliminary analysis on the Philippines 2017 panel, and then there's been 2 analyses on the Vietnam panel, one in the current project and one in the an earlier project. And you can start to see some regions of chromosome 1, 6 and 11 that seem to be in Vietnam that seem to be associated with silicon, and again 1 and 11 in the Philippines. So there's some consistency about which genes seem to be important, and our current understanding is that our top pick, as it were, is a glycosyl transferase, so something that's associated with cell wall structure. So that one is being used in a gene editing test to sort of produce knockout mutants and see if that really is the responsible gene. What's interesting is that none of the transporters, well, there's LSI6 is on chromosome 6, but this variation is not associated with variation in the transporters at all. They seem to be very highly conserved across all the panels that we've looked at. Okay, so next steps is to continue with the GWAS, as I say, try and identify some new genes and test them and breed those into elite lines. And I'm running out of time, but I just want to, in the last literally 3 minutes, tell you about yet another thing that silicon can do, if you're not impressed enough already. So, silicate weathering. Now, you may have heard of some geoengineering approaches to trying to mitigate climate change. I've told you about how we might adapt our crops to climate change by making them more tolerant to salt or drought, but how can we actually reduce CO2 in the atmosphere? How can we actually mitigate climate change? Well, we can tip a load of basalt onto the soil. Now that sounds a little odd, but that dissolves in acid rain, and then you get silicon runoff, and that's sort of a silicon and calcium runoff, and that goes into the shells of ocean plankton in terms of calcium carbonate. And the other good thing it does is it reduces the acidity of the oceans as well. So silicon can not only save our crops, feed the world, fight off herbivores, fight off pathogens, deal with salinity, deal with drought, but it's going to save the barrier reef while it's at it. So now you probably get from my tone that I was a little sceptical about this approach, I have to say, fessing up here. Now, Leverhulme Trust were not sceptical, at least they were sufficiently unsceptical to give David Beerling at Sheffield 10 million quid to test it out in the Leverhulme Centre for Climate Change Mitigation. Now, David's looking slightly anxious here. I do wonder if he thinks, is this going to work? I might have to give him 10 million quid back. And this is Emma Bailey, who's a student with David and Euron Ton and myself. She's full of the eager optimism of youth, like you guys, and thinks, of course this is going to work. So what she did was she applied basalt to maize plants. Here's her maize plants. And she had control plants, and then she added basalt and let them grow and measured the silicon. And, well, I had to eat my words fast. So these are the control plants in green, and when you add basalt, the maize does take up more silicon. So the idea here, of course, is that if you apply basalt to, say, coastal crop areas, you get the runoff you need for CO2 drawdown from the atmosphere and ocean acidification, etc. And you also get a second benefit. You get improved food security because your crops have more silicon in. So the other thing she noted, of course, is that the older leaves have more silicon in than the newer leaves because silicon accumulates over time. So that's why you see these differences here. Okay, so then she said, well, does this work in terms of protecting the crops against herbivores and pathogens? She's just starting her herbivore experiments now, but she's done some pathogen experiments already with northern corn leaf blight. So this is the hyphal length on the plants that had were basalt treated versus controls. And the hyphal length is reduced, and the numbers of hyphae per spore are also reduced. Slightly need the eye of faith here, but that's what her results showed. So it looks as if that adding basalt does a lot of good for crops. You've just got the slight issue of the CO2 energy required to grind up the basalt, but that's another story. So take-home message. I hope you've seen from this talk that if we're going to tackle these huge issues we're facing, climate change, increased demand for energy, increased demand for food, increased demand for water, we're going to need some interdisciplinary teams. Plant science is at the heart of it, but it's through working with other disciplines that we're going to be able to make a real difference. We need international collaboration. We have to come together and work across countries, particularly in countries where these issues are really going to be a problem. And we have to show real innovation and skill if we're going to deliver a change, real-world impact. But we've always done that. As human beings, we've been changing our farming from this to potentially something like this. Innovation is at the heart of it. Plant science is at the heart of it. And you guys are at the heart of it. This is what you can do through studying plant science. There's never been a more important time to be a plant scientist, to get out into the real world, to work with other people from other countries and other disciplines and fix this shit. Okay, thank you.